Okay, let's discuss how epithelia can be classified. There are more ways how to do that. The first would be according to their spatial arrangement, while the most common situation in the human body is that the epithelial cells are covering, protecting, and lining some surfaces or cavities. We call that covering or lining epithelia. An example could be epidermis, covering the human skin, or the mesothelium covering the body cavities such as abdominal cavity. Another case is when the cells are arranged in trabecules that are branching or anastomosing with each other in 3 dimensions. So we got complicated 3D network of cells. That's something we call trabecular epithelium, and it occurs in liver epithelium, in adrenal cortex, or for example in endocrine pancreatic islets. Another situation, quite rare, is when the cells, they even don't look like regular epithelial cells because they form projections, and they are touching each other with these processes and projections, forming a spatial network. This is called reticular epithelium. And that reticular epithelium could be found only in the thymus, where it supports the infiltrating lymphocytes, and in the enamel organ during the embryogenesis of tooth. Let's discuss and let's focus the classification of covering epithelia, which are the most common in the human body. So if you encounter a covering epithelium, you will look on the number of layers, and there could be either one layer, which you call simple, or the number of layers equals or exceeds 2. That's what you call stratified. The second feature you'll look at will be the shape of the surface cells. There could be like 3 situations. First, the cells could be flat, only the region with the nucleus could be bulging. You call it squamous. Or the cells would be cuboidal with round-shaped nucleus, or the height of the cells would exceed their width, mostly with ellipsoid nuclei. That's what you call columnar. Now we combine both these criteria to get the full name of the covering epithelium. So the first combination would be simple squamous, right? The simple squamous epithelium consists of a single layer of flat cells on the basal membrane, and it's lining the mesothelium of serous cavities such as peritoneal, pleural or pericardial cavities. It lines the lung alveoli in the form of type 1 pneumocytes. It lines the outer layer, you call it parietal layer of the Bowman's capsule of renal corpuscle. In the kidney cortex, or the thin segment of the Henle's loop, which is in the renal medulla. Okay, another combination would be simple cuboidal. So we got one layer of cuboidal cells, this epithelium lines the thyroid follicles, the follicles of the thyroid gland, or the sweat glands, or the distal canal, or the tubule of the kidney nephron. Another combination would be simple columnar. So we got a single layer of columnar cells, please note that the shape of the nucleus corresponds with the shape of the cells, because you mostly don't see the cell borders, but you always clearly see the nuclei, so it's very helpful in the microscopy. And this epithelium lines the large and small intestine, so these are enterocytes of small and large intestine. As the stomach is lined with simple columnar epithelium, and also the gallbladder is lined with simple columnar epithelium. Outside the GIT, a good example would be the oviduct, the fallopian tube, and the uterine cavity. Other epithelia would be pseudo-stratified, columnar. Now the situation is somehow different from the simple columnar. In this case, you got the basal membrane, and you got short cells and tall cells. So there are nuclei in various distance from the basal membrane, but all the cells are touching the same basal membrane. So in the microscope you can see that this distance is definitely different than that one, which would suggest there might be more layers. But actually it's not true, because separate layer would require the top cells sitting on the bottom cells. But if all these are touching the same basal membrane, you call it pseudo-stratified. Pseudo means it would appear as if it were stratified, but it's not truly. This epithelium can be found in the nasal cavity, in the nasopharynx, trachea, bronchi, or in the epidermic duct, or in the ductus deferens. Another special epithelium is called transitional epithelium or urothelium, because it occurs only in urinary passages. It occurs in two shapes, two forms. Let's consider a dilated urinary bladder and contracted. And a stretched and relaxed urinary bladder, the epithelial cells seem to be flat, and the cells in all cells are touching the same basal membrane, although their feet are so thin that you would need an electron microscope to prove that. Yeah? So again, we might suggest as if it were stratified epithelium, but actually the cells are touching the same basal membrane. The cells on the top have very often thickened plasma membrane because it's folded many times, and it resembles an umbrella. That's why they are called umbrella cells. While a contracted urinary bladder looks a little bit different. The cells are taller, they are sliding over each other. And it's the same epithelium. What makes this even more complicated is the fact that the oldest generations of umbrella cells are gradually losing their feet, so they are losing the contact with the basal membrane. This occurs in the renal pelvis, including its branches known as the minor and major calyces. It occurs in the ureter, urinary bladder, plus the proximal segment of the urethra. Other epithelium would be stratified squamous. Now we come to stratified epithelia, okay? So stratified squamous, which occurs in two forms in the human body. First, as a keratinized or non-keratinized. I will start with a non-keratinized, actually. Where the cells in the basal layer are the cuboidal, And you got layers with cells of a polygonal shape linked with desmosomes. And gradually the cells become more flat, more squamous. But even in the top layers, they retain the nuclei, their nuclei. So if you look on the top layers, they still have nuclei, because these are living cells with nuclei. You call it stratified squamous, non-keratinized. It lines most of the mouth cavity. Let's call it oral cavity, right? Pharynx. Oropharynx. Esophagus. Or vagina. However, another form of stratified squamous epithelium has some modification. Namely, the basal layer looks similar. Also, the spinous cell layer above looks similar, but then comes a layer with cells containing dark granules. These are granules of a substance called keratohyalin, and these keratohyalin granules will differentiate or mature into keratin, but the cells will die, they will lose their borders, and they will be gradually filled with a scleroprotein called keratin. You cannot exactly tell where the cell borders were, because they are filled completely with keratin. So this is the keratinized layer. Which is obviously made of dead cells filled with keratin and with no nuclei. No nuclei. Here are the cells with keratohyalin granules. So if you see something like that, it's stratified squamous keratinized. And this epithelium covers the surface of our skin in the layer called epidermis. Another epithelium would be stratified cuboidal. So usually we got two layers of cuboidal cells. It occurs, for example, in interlobular ducts. Of salivary glands, of large salivary glands, major salivary glands. And the last example would be stratified columnar epithelium, where even if the bottom cells, the basal cells, would be cuboidal, according to the naming rule, if the top cells are columnar, the whole epithelium is. So this kind of epithelium occurs in the anorectal junction of epithelia, where the simple columnar epithelium of the rectum joins the stratified squamous non-keratinized epithelium of the anal canal. So there is a short zone of this. Or there is a transitional zone on the epiglottis. Again, where the pseudostratified columnar ciliated epithelium of the epiglottis is merging with the stratified squamous non-keratinized epithelium towards the root of tongue, or the fornix of conjunctiva, of the palpebral conjunctiva. Okay, so these were simplified schemes that might help you when studying atlases and real slides. So good luck with that.